Welcome to our in-depth exploration of Chapter 3, Sterilization, Disinfection, and Antisepsis. In this chapter, we delve into the critical practices of infection control, vital for preventing healthcare-associated infections, HAIs. Let's break down the essential components of sterilization, disinfection, and antisepsis. Infection control is paramount in clinical settings to prevent HAIs. Sterilization, disinfection, and antisepsis are key practices that form the backbone of these efforts. Let's start with sterilization. Sterilization is the complete elimination of all forms of microbial life, including bacteria, viruses, fungi, and spores. There are three main types of sterilization: physical methods, gas vapor sterilization, and chemical sterilization. Let's discuss each in detail. Physical methods of sterilization. Saturated steam under pressure, autoclaving, the most effective and commonly used method, operating at 121 degrees Celsius for 15 to 20 minutes. It works by denaturing proteins and disrupting cell membranes through high heat and moisture. This method is used for sterilizing surgical instruments, labware, and biohazardous waste. Dry heat. This method is used for materials that can be damaged by moisture, such as powders and oils. It operates at 160 to 170 degrees Celsius for 2 to 3 hours, oxidizing cellular components. The high temperatures cause cellular oxidation, effectively destroying all microbial life. Filtration, used for heat-sensitive liquids and gases. This method physically removes microbes via membrane filters with pore sizes typically 0.2 micrometers. Applications include sterilizing culture media, intravenous solutions, and air in operating theaters. Filtration traps microorganisms, preventing them from passing through the filter. Radiation, ultraviolet UV light is used for disinfection rather than sterilization and is limited to surfaces and air. Ionizing radiation, such as gamma rays and X-rays, is used to sterilize disposable medical equipment and pharmaceuticals. This method damages DNA and cellular structures, preventing microorganisms from replicating and causing cell death. Gas vapor sterilization methods. Ethylene oxide, ETO, effective for heat and moisture-sensitive materials. It operates at 30 to 60 degrees Celsius for 2 to 5 hours in a controlled humidity environment. ETO alkylates proteins and DNA, disrupting their normal function and preventing microbial replication. It's used for sterilizing medical devices and electronics. Hydrogen peroxide vapors. This method includes vaporized hydrogen peroxide, VHP, and plasma gas sterilization. It produces reactive oxygen species that damage cell components. These reactive molecules attack proteins, lipids, and DNA, leading to cell death. These methods are used for endoscopes and surgical instruments. Chemical sterilization methods. Peracetic acid, used in automated machines for endoscopes. This chemical disrupts cell walls and membranes and oxidizes proteins. Its potent oxidizing action ensures complete microbial destruction. Glutaraldehyde. This chemical requires prolonged exposure for sterilization. It alkylates amino and sulfhydryl groups of proteins, leading to cross-linking that inactivates vital microbial functions. It's used for sterilizing medical and dental equipment. Now let's move on to disinfection. Disinfection is the elimination of most pathogenic non-spore-forming microbes from inanimate objects. Disinfection processes are categorized into high-level, intermediate-level, and low-level, each with specific applications. Table 3.2 provides a detailed summary of various disinfectants, their concentrations, and levels of activity. High-level disinfectants approach sterilization, killing all microorganisms except high numbers of bacterial spores. Agents include moist heat, glutaraldehyde, hydrogen peroxide, peracetic acid, and chlorine compounds. These are used for semi-critical items like endoscopes. Intermediate-level disinfectants kill mycobacteria, most viruses and bacteria, but not bacterial spores. Agents include alcohols, 70 to 90% isopropanol, iodophore compounds, and phenolic compounds. These are used for non-critical instruments like stethoscopes and thermometers. Low-level disinfectants kill some viruses and bacteria, but not mycobacteria or spores. Agents include quaternary ammonium compounds, which are used for environmental surfaces such as floors and walls. Factors influencing disinfectant effectiveness include the nature of microbial contamination, the number of microorganisms, bioburden, the presence of organic material like blood and pus that can inactivate disinfectants, the type and concentration of disinfectant, the duration and temperature of exposure, and compatibility with the surface or instrument being disinfected. Antisepsis involves applying chemical agents on skin or other living tissue to inhibit or eliminate microbes, excluding spores. Table 3.3 provides details on common antiseptic agents, including their concentrations, mechanisms of action, and applications. Let's explore some of these antiseptics. Alcohols, such as ethanol, 60 to 70%, and isopropanol, 70 to 90%, denature proteins and disrupt cell membranes. They are fast-acting and leave no residue, but can dry the skin and are flammable. Iodophores, like povidone iodine, penetrate cell walls and disrupt protein and nucleic acid structure and synthesis. They offer a broad spectrum and good residual activity, but can cause skin irritation and staining. Chlorhexidine, often used as a surgical scrub, disrupts cell membranes and precipitates cellular contents. It has broad activity and persistent action, but is inactivated by organic matter and can cause allergic reactions. Perachlorometaxylenol, PCMX, has limited efficacy against gram-negative bacteria and is used in hand-washing products. Triclosan inhibits fatty acid synthesis and is found in deodorant, soaps, and toothpaste, though there are concerns about resistance and environmental impact. Mechanisms of action. Moist heat coagulates and denatures proteins. Autoclaving is the standard method due to its effectiveness and reliability, ensuring that all microbial life is destroyed through protein denaturation and membrane disruption. Ethylene oxide alkylates proteins and nucleic acids, preventing microbial replication by adding alkyl groups to molecules, which disrupts their normal function. Aldehydes, such as formaldehyde and glutaraldehyde, cross-link proteins and DNA. This cross-linking action makes it difficult for the microbes to replicate and repair, leading to their death. Oxidizing agents like hydrogen peroxide and peracetic acid generate free radicals that damage cellular components. These reactive oxygen species attack essential molecules within cells, leading to broad-spectrum efficacy, including the destruction of spores. In addition, the chapter provides a glossary of terms that summarizes the most important concepts. Some examples already mentioned before and that you can review in this chart include sterilization, disinfection, antisepsis, and other terms such as high-level disinfection, intermediate-level disinfection, low-level disinfection, and so on, that you can take a look. In conclusion, this chapter emphasizes the importance of appropriate sterilization, disinfection, and antisepsis methods to prevent infections, particularly in healthcare settings. Understanding the mechanisms, applications, and limitations of various methods ensures effective infection control and patient safety. Thank you for joining us in this detailed exploration of sterilization, disinfection, and antisepsis. Stay tuned for more informative content not only in microbiology, but also in other branches of medicine. If you found this content useful, please like the video, share it with others, and subscribe to our channel. Don't forget to activate the notification bell to stay updated on all our latest content.